All right, welcome back to The Explainer. Today we're diving straight into what I honestly think is one of the most baffling mysteries in modern longevity science. We're always on the hunt for the Holy Grail, right? That one single compound that can reliably add healthy years to our lives. But what happens when the data throws us a massive curveball? What happens when a potential fountain of youth works incredibly well, but literally only for half the population? Let's unpack an anomaly that is completely forcing researchers to rethink mammalian aging.
Let's kick things off with a pretty wild question. What if a variant of the female sex hormone was actually the ultimate biohack for male longevity? I know, it sounds totally counterintuitive. Estrogen is, fundamentally tied to female biology. But imagine a male suddenly getting this massive biological upgrade from an estrogen-related compound. It sounds crazy, but trust me, the data here is incredibly robust.
We aren't just talking about theoretical science fiction here or some tiny statistical rounding error. Rigorous testing actually shows that this specific estrogen-like compound bumps up the median lifespan of male mice by an absolute astonishing 19%. To put that into perspective, in human terms, that'd be like reliably adding a decade, maybe even more, of completely healthy life. A 19% median lifespan extension is literally a towering achievement in longevity research. I mean, most compounds we test don't even move the needle a single percentage point.
Now you might be wondering, how can we be so absolutely sure about this 19% figure? Well, we have to look at exactly where this data is coming from. It's from the National Institute on Aging's Interventions Testing Program, or the ITP. For those of us following the science of aging, you already know the ITP is the absolute gold standard for reproducibility. They don't just run a test in one lab with one breed of mouse and call it a day. They use genetically heterogeneous mice to actually mimic human genetic diversity, and they run these experiments simultaneously across 3 totally independent testing sites. We're talking the Jackson Laboratory, the University of Michigan, and the University of Texas. Basically, if a drug extends lifespan in the ITP, you can take that straight to the bank.
So, since they started back in 2002, the ITP has been systematically hunting for real anti-aging shortcuts. They're basically the ultimate biological lie detector for all those hyped-up supplements you see online. But it actually wasn't until 2014 that they published their initial, groundbreaking findings on our mystery compound. And just to ensure absolute, rock-solid rigor, by 2019 they even added a dedicated data coordinating center. Look at it this way. Out of thousands of molecules that people theorized could slow aging over the last couple of decades, the ITP has only formally validated around 15 specific agents, plus a couple of combinations that actually work. Finding one is rare enough, but finding one that acts the way this one does, that is practically unheard of.
Okay, so what exactly is this mystery compound? It's called 17-alpha-estradiol. Basically, it's an isomer, think of it as a structural cousin, of the primary female sex hormone, 17-beta-estradiol. But here is the absolute crucial distinction. It is non-feminizing. It's roughly 100 times less potent at binding to classic estrogen receptors. So what does that actually mean? It means when it's given to males, it doesn't induce female sexual characteristics. You get all the hidden biological benefits without any of the feminizing side effects. It operates almost like a stealth molecular agent inside the male body.
And here is the massive paradox that completely shocked researchers. They administered this compound, right? They were fully expecting just a broad general anti-aging effect across the board. But instead, the male mice experienced up to that massive 19% median lifespan extension we talked about. And the female mice? 0%. Literally zero. They saw absolutely no lifespan benefit whatsoever. It was basically like the females were taking a sugar pill, while the males were taking a literal youth elixir. Crazy, right?
The deeper you go into the details of this data, the more incredible it gets. Not only does 17-alpha-estradiol extend the median lifespan, but it actually increases the maximal lifespan too, meaning the absolute longest-living top 10% of males lived even longer. Now normally, female mice naturally live longer than male mice, which is super similar to humans, but the males taking this compound actually end up completely outliving the untreated females, erasing that natural female survival advantage entirely. And honestly, perhaps the best news of all, it's highly effective even when you start it in late middle age, so the human equivalent of around 50 or 60 years old. You don't have to be taking this from birth to see the benefits.
Now, if 17-alpha-estradiol was the only drug that did this, we might just shrug and write it off as some weird quirk of that specific molecule. But cue the plot twist. It turns out this isn't some isolated anomaly, not at all. It's actually part of a massive, hidden pattern in longevity science that researchers are honestly just now beginning to fully acknowledge.
When we start looking at other highly successful drugs validated by the ITP, we see the exact same towering male-only lifespan extensions. Take acarbose, for example. It's a pretty standard diabetes medication, but it extends male lifespan by up to 20% while having a tiny, almost negligible effect on females. Or look at canagliflozin, another glucose-regulating drug. That one boosts male lifespan by 14%, with, you guessed it, absolutely zero benefit to females.
The numbers perfectly illustrate just how massive this biological divide really is. While some interventions, like the famous mTOR inhibitor rapamycin or the amino acid glycine, work beautifully across the board for both sexes, a staggering portion of our absolute best longevity drugs, like 17-alpha-estradiol, canagliflozin, and NDGA, are completely 100% male-biased. Out of the 14 major compounds the ITP has found to increase lifespan, 8 of them work predominantly or exclusively in males. And get this, none of them work exclusively in females. Not one. So, that brings us to the absolutely crucial question, the why. Why on earth are so many of these successful longevity molecules working exclusively in males? What exactly is it about male and female biology that makes them react so drastically differently to the exact same anti-aging interventions? Why is mammalian aging just fundamentally different depending on your biological sex?
Well, one major clue actually comes from how these drugs operate metabolically. Remember acarbose and canagliflozin? Both of these drugs do essentially the same thing. They blunt postprandial glucose spikes. Basically, they stop your blood sugar from shooting up right after a meal. The fact that blocking those glucose spikes extends male lives so incredibly effectively, but does practically nothing for females, suggests something pretty profound. It looks like male biology is significantly more vulnerable to the systemic, long-term damage caused by high blood sugar. Females, on the other hand, seem to just have this innate biological armor against this specific type of metabolic wear and tear, an armor that males simply lack.
And this realization, it is sending shockwaves through the scientific community. As Dr. Nirbhar Zyla, who is an absolute legend in longevity research, points out, science has historically just ignored these vital sex differences. For decades, researchers relied predominantly on male mice for testing, assuming the results would just seamlessly translate over to females. But the molecular data we have right now is forcing a total paradigm shift. We have to acknowledge that aging isn't just a singular, universal pathway. It is highly, highly dependent on your biological sex.
And we can actually see this difference playing out at a microscopic cellular level. When researchers zoom in and look at the proteome, which is the entire set of proteins expressed by an organism, as it ages, the divergence is just stunning. In aging males, there are over 600 proteins that significantly increase. They basically go haywire as the body gets older. But in aging females, that number is less than half. Females just maintain a highly stable biological environment, a stable proteome, well into old age. Males, at a molecular level, are just far more chaotic as they age, which, honestly, might be exactly why these radical chemical interventions, like 17-alpha-estradiol, can swoop in and provide such massive stabilizing benefits just for them.
So, what should we actually take away from all this data? Well, for anyone serious about health span and anti-aging, the path forward really requires a shift in how we think. First off, we absolutely must acknowledge these deep, foundational differences in aging biology. Second, as future therapeutics start hitting the market, we have to start tailoring interventions and dosages specifically by sex. And finally, we have to completely abandon that outdated one-size-fits-all approach to biohacking. Because clearly what works absolute wonders for a man might do absolutely nothing for a woman, and vice versa.
As we continue to unlock all the secrets behind the 17-alpha-estradiol paradox, it leaves us with this incredibly thrilling, provocative thought. If we've been getting something as fundamental as aging so incredibly wrong for so long just by ignoring sex differences, how is this revelation going to rewrite the rules of modern medicine for everyone? Imagine a future where literally every treatment, every single longevity protocol is biologically tuned to your exact genetic and hormonal makeup. That isn't just the future of anti-aging, that is the future of how we understand human life itself. Thanks for tuning into this explainer and keep questioning the science.